For decades, we really thought we had the upper hand. Antibiotics were our miracle weapon against bacterial infections. But here's the thing, the enemy is evolving. We are facing a silent pandemic, one where our best defenses are starting to fail. This is the story of antimicrobial resistance, or AMR, and how we're fighting back by, cracking the enemy's code.
This is a video generated to support Dr. Ramy Aziz's presentation at Faros University's Pharmacy Forum on April 18, 2026. Feel free to watch the video as many times as you want, as it highlights in more details some specific aspects that were quickly covered during the forum.
Okay, so let's just dive right into the biggest question of all, the one at the heart of this entire explainer. Can genomics, the science of reading DNA, actually give us the edge we need to survive this antimicrobial resistance pandemic? Because believe me, the stakes could not be higher. So, here's how we're going to break it down. First, we'll get a handle on the sheer threat of the superbug pandemic. Then, we're going to completely flip the script and look at this whole thing from the microbe side. After that, we'll get into 3 really game-changing ways we can use genomics before we wrap it all up by looking at how we can actually win this biological arms race. First up, let's talk about the scale of the problem. And I want to be clear, this isn't some far-off, futuristic threat. No, the superbug pandemic is already here, and it's an absolutely massive challenge to global health. So who are the main culprits in this story? Well, meet the escape pathogens. And that is not just a clever name. It's a very real warning. These bacteria are masters at escaping our drugs. You've probably heard of Staphylococcus aureus, or its resistant form, MRSA. I mean, it can cause anything from a simple skin infection to deadly pneumonia, and it is incredibly tough to treat. These 6 right here, they're the leading cause of dangerous hospital-acquired infections all over the world. And these superbugs, they are expert travelers. Seriously, our totally interconnected world has become a superhighway for them. Just look at this case from 2013. A tourist gets hospitalized in Egypt, picks up a multi-drug-resistant bug, and brings it all the way back to the Netherlands. It's a pretty stark reminder that a superbug in one part of the world can become a global problem, like overnight. And this photo, this is from the Patterson case, and it just shows you the devastating human cost. This patient was infected with a pan-resistant bacterium. Pan-resistant means no known antibiotic could treat it. This is the reality of a post-antibiotic world, where a common infection can suddenly become a death sentence. So we've seen the human side of this fight. But to really understand what we're up against, we need to change our perspective, like completely. Let's try to look at this whole thing from the microbe's point of view.
Exactly. We call it a war, a conflict, a battle. But what if, from the bacterium's perspective, it's something else entirely?
See, for a microbe, becoming resistant isn't an act of aggression. It's just survival. It's evolution playing out on hyperspeed right in front of us. Every single time we use an antibiotic, we're creating a pressure cooker, a life or death test. The bacteria that just happen to have the right genetic tools to survive, well, they win. They divide, they take over, and they pass those winning tools on. Their entire playbook is written right there in their DNA. So if all their secrets are in their genome, well, then that's exactly where we need to look. Genomics gives us the power to read their code, to understand their history, their defenses, and, most importantly, their weaknesses. It is our key to unlocking the truth.
And that brings us to the first huge application of genomics in this fight, surveillance. I want you to think of this as our global intelligence network. It lets us track the enemy's movements in real time.
We call this smart epidemiology. So instead of just waiting around for an outbreak to happen and then reacting, we can get ahead of it. We can proactively sequence bacterial genomes from patients. Imagine this. Health officials spot a new resistant strain in one hospital. By instantly sequencing its genome and comparing it to a global database, they can see if it's popped up anywhere else, they can track its path, and they can warn other places before it becomes a massive crisis. It's a total game changer.
Okay, on to our second application, diagnostics. Once the enemy is inside the gates, so to speak, we need to identify it and everything it's capable of with incredible speed and precision. The old way of doing things, the traditional way, involved growing cultures in a lab. That could take days we just don't have. With sequence-based diagnosis, we can go straight to the source. By reading the pathogen's DNA, we can figure out exactly what it is, and crucially, we can see the specific genes it's carrying that make it resistant to our antibiotics. And we can do all of this in a matter of hours.
And a single genome gives us an absolute treasure trove of intelligence. It's incredible. I mean, beyond the thousands of basic genes, we can pinpoint the exact troublemakers. We can spot the 70 to 80 genes that we know are linked to antibiotic resistance, which tells doctors precisely which drugs won't work. We can also see its virulence factors. Those are the genetic tools it uses to actually make us sick. It's like having a complete dossier on the enemy, letting us pick the absolute best treatment right from the start.
So we can track them and we can identify them. What's next? Well, the third application is all about how we fight back. It's about using genomics to develop completely new weapons for our arsenal. A pathogen's genome isn't just a list of its strengths. It's also a map of its vulnerabilities. It's Achilles' heel. By analyzing that whole genetic blueprint, scientists can find genes that are absolutely essential for the microbe to survive. And if we can design a drug that targets and shuts down the protein made by one of those genes, we can kill the bug in a totally new way, completely sidestepping its old resistance tricks.
Now, this idea actually builds on some really foundational work, like this paper from 1999 by Sukharenko and his colleagues. They figured out that as bacteria adapt to us, their hosts, they can actually lose genes they don't need anymore. And that's a critical insight. Why? Because if we can figure out which genes are so essential for survival that the bacteria can't ever get rid of them, then we found the perfect targets for new drugs that will be much, much harder for them to ever evolve resistance to. So where does all of this lead? Well, it leads us to the final chapter of our explainer, reframing this whole challenge as a biological arms race, that for the first time, we actually have the tools to win.
You've heard that saying, history is written by the victors, right? Well, in evolution, that's not just a clever phrase. It is a fundamental truth. The organisms that survive, the victors, they're the ones that get to pass on their genes. And for billions of years, microbes have been the undisputed champions of adaptation and survival. But now, something has changed. For the very first time in history, we can read their playbook, their genetic history, as it's being written. Genomics gives us the intelligence to see their next move and the tools to develop our own new strategies. We are arming ourselves to make sure that humanity gets to write the next chapter. We've opened a whole new front in this ancient war. The microbe's entire history of survival is written in its genome. Now, with the power of genomics literally in our hands, the question isn't about them anymore. The question is, what will we choose to write in ours?